Good afternoon, good morning, everyone, depending on where you are. And welcome to this session on interpreting cancer clinical trials. And we are very privileged to have Dr. Bishal, who is going to be our presenter today. I'm sure many of you know Bishal, but I'll probably do a very quick introduction. Dr. Bishal Gyawali is a medical oncologist. He is an associate professor in medical oncology department at Queen's University. And as some of you know, he has written a lot of peer-reviewed paper in major high-impact journals. And with that background, he's going to be taking us through this session on how to interpret cancer clinical trials. This session was organized by Queen's University and Common Sense Oncology, which is a movement that advocates for research that has impact on the patient, and together with the University of Rwanda through the Medical Oncology Fellowship. So without further ado, I'm going to welcome Dr. Bishal to take us through this session. Bishal.
Thank you very much, Fidel, for the kind introduction. Welcome, everyone. It's a big pleasure for Common Sense Oncology to partner with University of Rwanda. Fidel, as you all know, is a member of Common Sense Oncology, is an active member of Common Sense Oncology. And we are very pleased to see that Fidel has started this first fellowship program in Rwanda in oncology. And it's a matter of big pleasure for us to support this program. But I'd also like to welcome everybody else who is joining today. We wanted to extend this to everybody in the world who wanted to join this session and hopefully share some ideas about how to interpret phase 3 cancer clinical trials. So welcome, everyone. Okay, so a couple of disclaimers is that I'll be giving several examples, real life examples when illustrating a point, but those are not meant to be criticism of that particular trial or the set of authors who did the trial, but it's more for a learning point. So don't take it personally if you're involved in those particular trials that I criticize. These are to make a general point about how we should be interpreting clinical trials. So there are a couple of things that I do even before I start reading the paper. That includes, I look at who funded and sponsored the trial, what are the conflicts of interest of the authors of the trial, and who wrote the paper. Now, who wrote the paper might sound funny because, of course, there are a list of people in the authorship of the paper that you would assume have written the paper, but that's not always the case, interestingly. As you can see, in this particular paper, it says the company was responsible for collection, analysis, and interpretation of the data. I have no issues with the company helping out with or partnering in collection of data. Even analysis of data might be okay, but interpretation, I think the interpretation has to be done by the trialists or the authors, the academic authors who have supposedly written the paper. The interpretation should be left to them. But then, now this is very common. This is not a rare example. This happens in almost 90% of the papers that I read in these major journals. The manuscript was written with medical writing support, which was funded by the industry. So that's why I mentioned that part about who wrote the paper. Ideally, the authors will write the paper, but it seems like nowadays, the authors actually don't write the paper. The paper is written by professional medical writers who have been funded by the industry, and which is a problem because they have so much financial stake into the interpretation of the trial. And that's why it matters. To give you an example, this is the conclusion of an abstract. And if you can read this conclusion, there is so much of spin in this one paragraph in the conclusion of the abstract. So it says, although hazard ratio for overall survival was in favor of the drug versus placebo, although the hazard ratio was in favor, there was no statistically significant difference. If I had written this paper, if the industry's professionals had not written this paper, we would have just said the overall survival was not improved. It was not different. But it says, although hazard ratio was in favor, There was no statistically significant difference. In a random surrogate, PFS2 showed a clear trend for treatment benefit, but was not alpha protected. In simple terms, it means even PFS was not improved. But if you read this conclusion, you would not notice that this drug failed in the trial, and this trial was pretty negative even for OS and PFS. So watch out for abstract conclusion. Maybe you don't even need to read the conclusion of the abstract. There are only a couple of occasions in which I read the abstract, but the conclusion of the abstract, I think you never need to read that. You can interpret it yourself. So don't rely on the abstract conclusion. And be very aware of experts, because in our field, we give very much importance to supposedly what we call key opinion leaders or the KOLs. But key opinion leaders are not necessarily always the best person to interpret the trial results. They always have conflicts, and they interpret the results that suit their financial or personal agenda. And the thing is, because we think the experts are real experts, no matter what they say, we can interpret it in a different way. Or even the experts themselves, they can interpret it whatever way they want, irrespective of what the data shows. As I showed you in the previous example of that abstract conclusion, even if the trial was so negative, the conclusion was spun in such a way that it did not read negative. So, I find it fascinating. I read this in a news article that a pair of glasses were left on the floor at a museum and everyone mistook it for art. So you can interpret things whatever way you want. And especially if the KOLs are doing it, then you'll try to trust it more than needed. I'll give you another example. So this was a trial from 2016, a pretty old trial now. But this was the trial of a drug for cancer cachexia. And of course, cancer cachexia is a big deal, and we do not have good drugs to treat it. So this was a very welcome trial done in an otherwise neglected field. But let's look at the results. In these two trials, drug versus placebo, drug called enamorelin versus placebo, they randomized patients into two groups, and then they looked at the outcomes. One was how much skeletal muscle mass did the patients gain. And if you look at the muscle mass gain, if you get the drug, you gain 0.9 kilograms. And if you get placebo, you lose 0.47 kilograms. In the other trial, you gain 0.65 kilograms versus losing 0.98 kilograms. So in both cases, the difference is like 1 kilogram or less of muscle mass. Is it clinically meaningful? That's the question. But it is statistically significant. As you can see, the p-value is less than 0.0001, which is highly statistically significant. And we'll talk about that later on. But is it clinically meaningful? Do you think it matters whether your skeletal muscle mass is 1 kilogram more or less? What matters is function, whether that difference in muscle mass allows patients to do more things than what they could have done. And they actually tested for this. And they tested it in terms of hand grip strength, which is a surrogate for muscle function overall. So if you look at hand grip strength, then there is no difference. It was not even statistically significant. So what this basically means is if you get this drug versus not getting the drug, then you will get like 1 kilogram extra muscle, but your ability to button your shirt or open the door handle does not change. So what meaning is that? So if you look at the results, then you will conclude that this drug is not useful. Like, what's the point? But that's not what the conclusion says. The paper's conclusion says that considering the unmet medical need, enamorelin might be a treatment option for patients with cancer, anorexia, and cachexia. It does not say that enamorelin is not useful. It says it might be an option. And you would wonder why they would conclude that way. And this explains, of course, this paper was interpreted and written by the sponsor, the drug company. And that's why it's a problem. And that's why I'm telling we need to look at who wrote the paper. And then the question about experts. Okay, you would think that, okay, this paper was written by the company and therefore they concluded it in a very favorable way. But all these big papers, they come with an accompanying editorial, and that editorial is supposed to put things into proper perspective. So you would think that the editorial at least will say that this drug is not useful. And that's what I expected when I read the editorial for this paper. But the editorial concludes that these trials offer new hopes for more effective therapeutic strategies for cancer cachexia, and it should be integrated to produce measurable effects on patients' quality of life and clinical outcomes. So the editorial is saying that these trials are offering new hopes for patients. The editorial is super favorable. And I thought, like, why would the editorial be so favorable? Of course, the paper is favorable because the industry wrote it. But editorial, why is this editorial not concluding uh, correctly? And then I looked at the person who wrote the editorial, and the person who wrote the editorial has received lecture fees as well as advisory board member fee from the exact same company that made this drug. As you can see, it's the same company that made the drug, and the person who wrote the editorial has received money from the exact same company. Of course, it would be favorable. And I thought, like, is this a one-off thing or is it common? So we looked at this systematically and we published these results, and we found that if the editorialist is receiving money from the exact same company, which we call as a direct financial conflict of interest here, then it is very likely that the editorial will be unduly favorable. So if you're receiving money from that company, you'll write a very favorable editorial for the given product. So we have now empirically documented this. So take-home message for us is all these experts or key opinion leaders who write editorials or who write commentaries or who do discussions of the plenary sessions or the discussion of the presidential sessions at ASCO, ESMO, these major meetings, we need to look into the conflict of interest. And if we are organizing a conference, we should make sure that the discussion should not have conflict of interest, at least with the exact same company whose drug is being discussed. Or if we are in the journal, then we should not be asking editorials from these people. But if we are an audience, then we should at least, before trusting everything that these KOLs are saying, we need to check the financial conflict of interest and then take everything that they say with a grain of salt. Okay, these are background. These are the things that I do even before I read the paper. Now going into the paper, methods or results. So this is the most crucial part. And in fact, we have published a paper 3 years ago about different biases that can creep into study design, implementation, and analysis of cancer clinical trials that can affect our interpretation of the data. And I would highly encourage you to read this. We outline 10 biases that can creep into the trials and how to address them. But I also want to let you know that CSO is working on a checklist that you can use to go through any cancer clinical trial, especially drug trials, and see whether certain biases exist or not. So the CSO RCT checklist is coming out pretty soon now. So stay tuned, and when this checklist is out, we'll send you an email as well. And please start using the checklist whenever you are reading any oncology drug trial, so that we can arrive at correct interpretation. Okay, some of the methodological items that we need to be careful about. First, RCT versus single arm trials. And why is this important? Because randomization allows you to answer the question, what would happen if not for the intervention, right? And that's the question that we need to answer in clinical practice every day for the patient. We offer them different treatments, and the question for the patient is, what if I don't get this? What if I do nothing? Or what if I get the other treatment? What happens in that counterfactual world where you don't pursue that treatment versus pursuing that treatment? That question can be answered only through a randomized trial. A single arm trial will not tell you what might happen if you did not get the treatment. So therefore, there is the importance of randomized trials. Single arm trials are biased because of lack of randomization. You are selecting a selected population. So it's a highly selected group of patients that you enroll. And it's not powered, and there is no good criteria to define success. In oncology, if you look at the cancer drugs that have been approved in recent years based on response rate from single arm trials, at least in randomized trial, you can look into statistical significance and clinical meaningfulness. But in single arm trial, how do you define success? We have drugs that have been approved on the basis of 20% response rate in 50 patients. And then there are drugs that have 35% response rate, but only in 30 patients, 50% response rate in 40 patients, 17% response rate in 200 patients. How do you define success? There is no hard and fast way to define success in single arm trials. And single arm trials are actually not meant to inform practice. And the other problem is the response rate and duration of response that you see in single arm trials are usually exaggerated compared to the same drug in the same setting in a randomized trial because of highly selected population, or also because of a regression to the mean effect because the population is small in single arm trials. In this paper, now several years ago, they looked at the chemotherapeutic drugs and their response rate in phase 3 studies versus phase 2 studies. And as you can see from this graph, the response rate in phase 2 or single arm trials is almost always systematically higher than the response rate that we see in phase 3 trials. We studied the same thing for so-called targeted drugs or biomarker-based drugs, and we found something similar. And not only for response rate, but for duration of response. We found that the duration of response is also almost always exaggerated in single arm trials compared to randomized trials. So when we are looking at results from single arm trials, we need to keep in mind that the responses from single arm trials are usually inflated. So if the response rate is 40%, then in the real world, probably it's much less than that. But what's the role of single arm trials? As I said, it was not meant to inform clinical practice. It was meant to inform whether or not a phase 3 randomized trial should be done. And in fact, we looked at a number of phase 3 trials that had failed, so-called negative phase 3 trials. And we found that 42% of the negative phase 3 trials, they did not have any phase 2 trial evidence, and only 28% had a positive phase 2. 14% of negative phase 3s were conducted despite a negative phase 2. So what is happening here is we are supposed to do a phase 2 trial, and if the signal was positive, we are supposed to do a phase 3 randomized trial. But we are doing phase 2 trials, and we are approving drugs based on phase 2 alone, without asking for a phase 3. And on the flip side, even when phase 2 is negative, we're still doing a phase 3 trial, even when phase 2 is negative. As in this paper we saw, there were 14% of phase 3 trials that were negative that were conducted despite a negative phase 2. And then we end up with a negative phase 3. A negative phase 3 is a disaster in all sense. It's a financial disaster because it costs a lot to do a phase 3 trial. But it's also a bad news for the patient. It's a bad news for the whole community because, huge amount of resources, including patient resources, were used in a trial that ultimately ended up being negative, when in fact the same resources could have been used in a trial that had some signal that it might be beneficial. So conducting a phase 3 when the phase 2 is already negative is not the right thing. So what that means for us is if it's a phase 3 trial, then we need to check, especially in the introduction or the generally in the introduction section or the background section, they mention whether a phase 2 was done or not and what the results of that phase 2 was. Then in terms of eligibility criteria, the question that we need to answer when reading the paper is, are the patients in these trials representative of a routine patient population? And that's an important question to ask because of our strict eligibility criteria. We know that the patients who are part of the trials are usually not patients that we see in the routine clinic. And what do we mean by that? For example, ASCO had released this statement about how to broaden the eligibility criteria. The average number of eligibility criteria to get enrolled into the trial is 16, and 60% of them are related to comorbidities. And ASCO has made this statement asking specifically that the trials do not exclude patients with brain metastasis that are stable or treated routinely from trials, or based on age, or based on renal function, or based on liver function. And it's In a sense, it feels like the patients who fit all these inclusion criteria and get enrolled into the trials are the fittest among patients with cancer. But in routine practice, we see patients with so many comorbidities, so that the results of the trial may not be generalizable to the patients that we see in routine clinic. In this paper that we looked at whether the patients in the phase 3 trials that have led to cancer drug approval in the US are representative of the US patient population. And we saw that female patients, Black patients, patients with hepatitis infections, and patients with brain metastasis, they were systematically very underrepresented compared to the normal patient cohort that we see in routine practice. And why that matters, I'll give you one specific example. For example, let's talk about sorafenib in advanced liver cancer. This was the first drug to be approved in advanced liver cancer, sorafenib, many years ago. I think this was like in 2005. As you can see, the control arm is placebo. And that's fine because at the time there was no other drug for advanced liver cancer. And this was the first pivotal trial to show overall survival benefit. As you can see, the placebo arm has a median survival of 7.9, almost 8 months. And the sorafenib arm has a median OS of 10.7, almost 11 months, so 3 months of difference, which looks good for a disease that does not have any other treatment option at the time. But in this paper, they looked at the real world effectiveness of sorafenib. What happens when we use sorafenib in the real world? And I don't want to go into the details of the methods of this paper, but they looked at the 60-day landmark as the benchmark because they wanted to exclude patients who die before that, because that could be for so many other different reasons. And then they looked at sorafenib versus patients who did not get treated. And they found 3 months versus 2 months. So if you include that 60-day, basically in simple language, what this means is patients who got sorafenib in the real world, they had a median survival of 5 months. And patients who got placebo had a median survival of 4 months. And that was mind-blowing for me, because as you can see in the trial, patients who got placebo had a survival of 8 months. In the real world, patients who got the drug had a survival of 5 months. That means patients who got placebo in the trial, they lived far longer than patients who got the drug in the real world. Now, why does that happen? Because the patients who got selected to participate in the trial had extremely good performance status, functional status, no comorbidities. And that's why they did far better than the drug did in the real world. And that's why it matters, the inclusion criteria. It leads to exaggeration of benefit. You can read this paper that I wrote about liver cancer, in which we compare the median overall survival for the drug versus the control arm. And as you can see, there are, like, even in second line trials nowadays, you see that the placebo arm has a survival of 8 months. We know that advanced liver cancer has a pretty bad prognosis. And at that time, when these trials were run, the median survival of advanced liver cancer in first line would be 6 months. Here in the second line, we are seeing placebo have a median survival of 8 months. That means these are hugely highly selected patients. These are patients with liver cancer, but one of the inclusion criteria is to have pristine liver function. Now you can imagine that. A patient with liver cancer, but we are expecting them to have good liver function to get enrolled into these trials. So these are highly, highly selected patients. So therefore, when we are reading the trial publications, we need to look carefully at the eligibility criteria and the so-called efficacy effectiveness gap. That means, do the results of this trial translate into the real world population? Then the other big thing to look into is the control arm. Is the control arm appropriate? And the control arm may not be appropriate in 3 different ways. One is if the trial is using a control arm that has already been proven to be inferior in previous randomized trials. If the trial is using a control arm that does not allow an effective drug, and I'll give you an example of that. And if the control arm is using placebo or ineffective drug instead of active or effective comparator. So using an inferior control treatment, we all know that for patients with non-small cell lung cancer with a PD-L1 of more than 50%, pembrolizumab is the standard of care. It has been the standard of care since 2016. Any patient with no issues of access should receive pembrolizumab. But this cemiplimab trial, it ran from 2017 until 2020 for the same patient population, and the control arm was chemo. In 2020, no patient with a PD-L1 of more than 50% would get chemo if access is not an issue. But this cemiplimab trial ran with a control arm of chemo until 2020. Of course, nobody would enroll patients into this trial in North America because patients would get immunotherapy anywhere, right, left, and center. So there is no point in enrolling patients in this trial. So this trial ran outside of North America, involving several countries in the Europe and other parts of the world. And they used that data to get the drug approved in the US. Now, there are several ethical questions about this, running a trial in low and middle income countries without guarantee of access to the drug and getting the drug approved back in high income countries. But if someone is running a trial of these new drugs, then at least they have to use the proper control arm. Because one of the principles of running a trial is no patient should be harmed as a result of participating in the trial. And we can easily argue in this trial that patients were harmed, patients who got randomized to control arm, because they would have received pembrolizumab if not for this trial. Or if we are talking about low-income countries where access to pembrolizumab was not possible, then at least there should be a guaranteed access to this drug even after completion of the trial, but there is not. And I think I'll have some data to show you about this in a couple of slides. The second type of not appropriate control arm is not allowing an effective drug. And what do I mean by that? For example, in this olaparib in metastatic breast cancer, the Olympiad trial, it says patients were assigned to receive either olaparib or standard treatment of physician's choice. Now, it does not sound wrong. Standard therapy of physician's choice seems like the right thing to do. But then there is a bracket, and the bracket limits the choice. It says capecitabine, eribulin, or vinorelbine. For patients with BRCA mutation, the physician might have wanted to use platinum. Maybe they wanted to use carboplatin. And the method does not say we prohibit people from using carboplatin, but it says we give physician choice, but the choice is among 3 options, which does not include platinum. So it's a fancy way of saying we are not letting physician use the best treatment. And the third is using a placebo control when placebo is not justified. There are occasions where placebo is justified, but using placebo control when an active treatment should have been used. So this is the POLO trial in pancreatic cancer. And as you may know, in the POLO trial, patients with advanced pancreatic cancer, they received FOLFIRINOX chemotherapy for 4 months. And they confirmed that patients were responding. Now, advanced pancreatic cancer is a pretty bad disease. It's a very lethal disease, bad prognosis. And if we give FOLFIRINOX chemo to patients and they tolerate it for 4 months and they respond to the treatment, then we are happy and we continue the treatment. If they are having some side effects with neuropathy, maybe we'll take oxaliplatin off, but we'll continue with the rest. But in this trial, after 4 months and confirming that the patients are responding, they randomize patients to get olaparib or placebo. Now think about that for a moment. Responding to chemo in a lethal disease like pancreatic cancer and getting getting placebo after 4 months. That is not reflective of the real world practice. And then they showed improvement in PFS. Of course, there will be improvement in PFS because the control arm got nothing, they got placebo. But even then, overall survival was no different, as you can see in the second slide. The curves are running together. So there was no benefit in overall survival. And what about quality of life? And they report quality of life as maintained. And quality of life maintained is a fancy way of saying quality of life was not improved. So basically, this is a drug that does not improve how a patient feels or how long the patient lives, but costs more than $12,000 a month and has important side effects and is approved. So this is where things are, and that's why we need to be vigilant about interpreting data. So the question is, who is going to control the control arm? We have written several papers about this. If you're interested, you can read them. This is about lung cancer. Should the control arms have an expiry date? This is about prostate cancer, because this is not an isolated issue for a couple of trials. This has been a problem in oncology for a while now. So going back to the question about people justifying using unethical control arm as saying, oh, but we are doing these trials in low-income countries where they don't have access to these drugs anyway. So the question is, can access to new drug in LMICs justify conducting a trial that would not have been considered ethical in high-income countries? And I think the answer is no, for two reasons. One, the Helsinki Declaration says that the control arm should be the best standard of care, not the local standard of care. But more importantly, these trials do not guarantee continued access to treatment after the trial is over. So in a sense, this is what research parasitism is, where we just use patients from low-income countries to get the drugs approved in high-income countries, which I think is unethical. And again, if you want to read in detail about this, I would like to refer you to this paper. But I'll give you some data to show how bad things are. In this paper, Evaluation of Drug Trials in High, Middle, and Low-Income Countries and Local Commercial Availability of Newly Approved Drugs, they looked at not just cancer, but any FDA-approved drugs whose trials was conducted globally, including outside of USA. And then they traced up to 5 years to see whether those drugs where these outside countries participated had access up until 5 years after the approval of the drug in the US. And this is what they found. Within 5 years, 46% of other high-income countries outside of USA had access to the drug. 9% of upper middle income countries and 22% of low income countries had access to this drug. So there is no access to these drugs in those countries that participated in the trial leading to its approval. And if you divide it by countries, and if we focus on Africa, this is the square block for Africa, and you can see that up until 5 years after approval, 0% of these drugs were available in Africa. So that means we're just using patients in Africa to do a trial that would have been unethical in North America and get the drug approved based on that data in North America, which I think is totally unethical. Okay, after that issue of control arm, the control arm issue does not end with what happens for the control arm patients during the trial. We should also focus on what happens after they progress. Okay, these patients got placebo, but when they progress, did they get the drug? These patients got chemo instead of pembro, but when they progressed, did they get pembro? So that is what we refer to as crossover. The control arm patients getting crossover to the interventional drug at the time of progression. But there have been several issues with crossovers. The crossovers are not ideal. Several of these trials, they in fact prevent crossover. They in fact prohibit crossover. And even when crossover is allowed, they don't make an effort to make sure that everybody gets crossover. It's up to the patients and the physicians to work hard to get access to the drug. So again, this issue, if you want to read in detail, I would like to refer you to this paper. There are several examples that we cite in this paper, because if you don't get crossover, for example, let's talk about drugs that are approved in second line or third line treatment, but now we're doing a trial in first line. Then the question there is not whether or not the patient should get the drug. Of course, we have already proven that it is effective in second and third line. The question is, should every patient get it first line versus only those patients who progress at second or third lines? And if you don't incorporate crossover, that question is not answered. The question is not whether this drug is useful. It is. Whether patients should get this drug at some time point versus whether they should get it upfront. That's the question that needs answering, and for that, crossovers should be included. So the take-home message for us is, first, we should look at the control arm and we should see, is this the standard control? Is this the treatment I would have loved to give my patients if there was not a trial? And second is, was crossover mandated or allowed or prohibited? And was there a justification why they allowed or prohibited crossover? And then there is post-progression therapies, other subsequent therapies, because in some cancers, we have 5, 6, 7, 8 lines of therapies now. Just to give you an example, in this trial of ribociclib in first line, this is a first line trial in hormone receptor positive breast cancer. Now you can see hormone receptor positive breast cancer will have so many lines of treatment, right? They have several lines of endocrine therapy. They have several lines of chemotherapy. They have several lines of HER2 directed therapy if they are HER2 positive. But I think in this trial, they were HER2 negative. But you have multiple lines of endocrine therapy and you have multiple lines of chemotherapy that you can give. This trial of ribociclib in first line showed that it improved overall survival, which is fantastic, improving overall survival. But if you look at the post-progression therapy, almost 25% of the patients, they did not get any subsequent treatment, which is not reflective of real-world practice. They did not get any treatment at all. Just think about it, having a hormone receptor positive breast cancer and having only one treatment option. You got first line treatment and that's it. There are no other several other hormonal treatments or chemotherapy options. And then if you look at the subgroup analysis based on geography, you see that the overall survival benefit is almost entirely driven from patients in Asia. And if you look at Europe and Australia and North America, then it does not look significant. So one can legitimately ask, did the Asian patients receive suboptimal post-progression treatment, that 25% who did not get any treatment, were they all entirely from Asia? And is that driving the overall survival benefit? That is plausible. I'm not saying that's the case. We don't know unless we can see all the data. That data is not provided, but it's plausible that OS gain may have been much smaller if the patients had received standard treatment, and if that 25% of the patients who did not get the standard treatment were all from Asia, and that subgroup is driving the entire benefit. So for earlier line trials, always examine data on post-progression therapies. Then comes the issue of the endpoint. What's the primary endpoint? So we have clinical endpoints and we have surrogate endpoints. Clinical endpoints are those endpoints that matter to the patients, like overall survival and quality of life. These are the things that the patients care about. Do I live longer? Do I live better? Ideally both. If not both, at least one. And anything else is a surrogate endpoint. Surrogate endpoints are things that the patients cannot feel or know themselves. Surrogate endpoints are those endpoints that we tease the patients about. So those are based usually on imaging. We do scans and we say there is a response or there is disease progression and so on. So if clinical endpoints matter to patients and surrogate endpoints actually patients can't feel, then why do we use surrogates in clinical trials? Because it saves time and allows earlier analysis and publication. For example, it takes a longer time to measure overall survival, but you can measure disease response and progression relatively earlier. But while saying this, I think people assume that by using surrogate, you save a lot of time compared to using overall survival. And I think that's not the case. You save some time, which is not negligible, but it's not like 5-10 years that people think it is. In fact, data has shown that compared to using OS, if you use response rate, you save on average 19 months. And if you use PFS, you save on average 11 months, which is not nothing, but it's not like 5-6 years that people think it is. But the problem is most of the surrogate endpoints are actually not good surrogates. What makes a good surrogate? If that surrogate endpoint is strongly correlated with overall survival and quality of life, then it is a good surrogate. But if that correlation is weak or poor, then it's not a good surrogate. And in fact, FDA says, now this is the language from the FDA itself. FDA says that clinical trials are needed to show that surrogate endpoints can be relied upon to predict or correlate with clinical benefit. surrogate endpoints that have undergone this testing are called validated surrogate endpoints, and these are accepted as evidence of benefit. But if you look at the validation of surrogate endpoints, then the validation in most cases is pretty low. The strength of correlation is very low. And therefore, when we're talking about surrogates, we can't put them all into the same bucket. We need to differentiate between actually validated surrogate endpoints versus non-validated, so-called putative surrogate measures. Just to give you an example, in breast cancer, we looked into the surrogate endpoints that the FDA considered acceptable, but even among the surrogate endpoints that the FDA considered acceptable, if we looked at the strength of evidence of correlation, then this is what we found. For pathological complete response, it was not at all correlated with overall survival. For response rate, it was not correlated with overall survival. PFS, only mild to moderate correlation, not sufficient. Event-free survival had some moderate correlation, but disease-free survival was not correlated with overall survival overall. But if you look at only the HER2 positive subgroup, then there was a strong correlation. So that's how we need to look at surrogate endpoints, depending on their strength of correlation in each individual setting. And when we make this argument about PFS not being a good surrogate for OS, people say, okay, it may not be a good surrogate for OS, but it should be a good surrogate for quality of life, because if the disease is not progressing, then you feel better. It's always good to hear that your disease is not progressing. So does that not matter in itself? And the answer is, of course, that's a fantastic question. And we need to answer that scientifically. And that's what we did in this paper. We wanted to answer, does PFS correlate with quality of life? Because that argument makes sense. But that argument forgets that PFS is not just quality of life is just not a function of disease progression, but it's also a function of drug toxicities. And if they balance out, then there will be no improvement in quality of life. So we wanted to test this empirically. And in this paper, we looked at correlation between PFS and quality of life. And we found very weak correlation between improvement in PFS and improvement in quality of life. So that was a good question to ask. But the answer is no, PFS is not a good surrogate for quality of life either. And in a more recent paper that we published in JAMA Oncology, we looked at drugs that have OS, PFS, and quality of life data. And we, as you can see, we found that for those drugs that improved overall survival, there was no case where it worsened quality of life, which is very reassuring. But there were several drugs that improved PFS, did not improve OS, and in fact worsened quality of life. So what are we to make of those drugs? We have drugs that does not improve survival, improves PFS, does not improve survival, but in fact worsens quality of life. So what are our patients getting from those drugs? And the other take home from this is, do not make quality of life conclusions based on PFS benefit alone. Just by seeing that PFS is improved, do not assume that quality of life has also improved. The trial should specifically measure quality of life as a secondary endpoint, pre-specify, and report it. We cannot assume quality of life has improved just by looking at PFS. Then the other thing to look into the results is the statistical significance versus clinical meaningfulness. So we all worship p-value of less than 0.05 and feel very happy about it. But as we discuss in this paper that I have cited here, there are several occasions where clinically the drug is pretty meaningless, but it will end up as statistically significant, and we should be aware of that. One example here is the NELA trial in HER2 positive metastatic breast cancer. Here, the median PFS is 5.6 versus 5.5 months. This is not a typo. This is actually the result. 0.1 month difference. But it is statistically significant. The p-value is 0.0059. And people are asking, okay, is PFS a valid surrogate for OS? I think you don't even need to ask this question. A 0.1 month difference in PFS is never going to translate into OS. But OS results were also already available, and OS, as expected, was not significant, of course. And this is how they concluded it. Although a numerical difference was observed for OS, the statistical significance was not reached. That means this was written by professional medical writers. So take home for us is don't believe claims of clinical or numerical difference in absence of statistical significance. If it is statistically not significant, then don't believe these claims of, oh, there is a numerical difference. But also, on the flip side, simply because there is a statistical significance, do not assume that it is clinically meaningful. As shown in this example, yes, p-value is 0.0059, but median PFS difference is 0.1 month, which is like 3 days, which is nothing. If you forget your scan and come for an appointment 1 week later, that will be a difference of 7 days. There are several other issues to discuss, but I think we are running out of time. There are issues about non-inferiority design trials, early stopping of trials, informative censoring, and subgroup analysis. I think we'll touch on these later in the future. But I want to tell you a couple of things about informative censoring because that's a new thing and many people are not aware. So censoring, what is censoring? Censoring refers to patients who do not complete the study and drop out without further measurements. Censoring we assume to be uninformative usually. That means people who drop out are balanced in both the arms, and that dropping out does not affect the results. But censoring can be informative if the dropouts are related to the treatment arm, and that leads to violation of the Kaplan-Meier principles or assumptions. And what do we mean by that? Let's assume that we have an experimental arm and we have a control arm. From the experimental arm who are getting the drug, what type of patients would drop out? Patients who got severe side effects, patients who became so sick that they could not come for follow-up, patients who died early. For so many reasons, there can be dropout from the experimental arm. So what type of patients are dropping out? Patients who are performing worse from the experimental arm are dropping out. But patients in the control arm, what type of patients are dropping out? Patients who got disappointed that they are in the control arm, they wanted treatment, and they are financially well off and educationally well off that they can seek this treatment outside of the trial, and they're going to other places to get second opinions and other treatments. So what type of patients are dropping out from the control arm? Best patients. So worst patients from the experimental arm and best patients from the control arm are dropping out. And that leads to informative censoring. And because after that, what we are essentially comparing, patients who are left in the trial, is the best performers in the experimental arm. We are comparing them with the worst performers in the control arm. And of course, we'll see an exaggeration of clinical benefit. Just to give you some example, these slides are from my colleague, Dr. Toma Meyerson from Israel. So I'd like to thank him for giving me these slides. So let's see control arm and intervention arm. Censoring is unrelated to outcome, and it is balanced between the study arms, that is uninformative censoring. So maybe these patients are censored. So similar patients, as you can see from both the groups, are censored. This is not going to affect our conclusions. But what is going to affect our conclusions is if this happens, the worst performing patients from the intervention arm are censored and the best performing patients from the control arm are censored. If there is a differential censoring, or yeah, so I'll give you this example. So simulation exercises have shown that, let's say we have a completely inert drug, a drug that does not work at all, hazard ratio of 1, p-value of 0.90. So this drug is completely useless. But if this drug is more toxic, and if there is just 5% more patients between the two arms that get censored, that alone, that censoring alone, can give you a hazard ratio of 0.75, which is statistically significant. So if you don't account for this, you would end up concluding that this is a good drug, when in fact it's a completely useless drug and even more toxic. So what percentage of the patients have been censored in the two arms? Is there differential censoring, and is that difference informative? Does that affect the outcome? That is something that we need to be very mindful about. Without looking into censoring, it's like this misleading picture. In this picture, you can see that someone is having a good time diving off the cliff, But if you look at the censoring, if you look at the entire picture, you'll see that it's actually not a good news. There is a shark waiting for this person underneath. So how can we address this? Ideally, they should have provided worst case scenario and best case scenario analysis accounting for censoring, but at least we need to see the reasons for censoring. Why were the patients censored? And we are encouraging at CSO for trialists to include this information in the supplement. Sometimes we can see that, sometimes we don't, but giving the reasons for censoring would be a big step. And finally, I'd like to talk about reporting harms more transparently in cancer drug trials. You'll see, especially in abstracts, that the harms are described as so side effects were acceptable, manageable, feasible. We should not get misled into this. So we should not allow the use of subjective downplaying terms like this. We should look into the objective toxicity data. The subgroup analysis and other, yes, for example, non-inferiority design analysis, we'll keep it for the next session. Yeah, I think we'll end it here today. So in summary, what you should look for in a clinical trial report: conflict of interest, abstract, who wrote the paper, is it a randomized or a single arm trial, is it a phase 3 or phase 2, what was the randomization allocation, what is the eligibility criteria, is the trial population representative, is the control arm standard of care, did they get crossover, was that appropriate, what are the post-protocol therapies, what's the primary endpoint, is it statistically significant, if it is it clinically meaningful, was the non-inferiority design justified, what are the basis for non-inferiority margin, was the trial stopped early, was the differential censoring, is the censoring informative, are the harms being downplayed, and so on. So thank you very much, everyone. I'd like to encourage you to follow Common Sense Oncology on our website as well as on Twitter. And I'm happy to take questions. I know we are running out of time, but I'm happy to stay a little longer to answer your questions. And I think the first priority for questions will be given to questions that come from the Medical Oncology Fellows at University of Rwanda.
Dr. Bishal, thank you so much for your time, and hopefully we shall get another time to go into this again, because we can't really discuss this in 1 hour. Thank you so much.
Thank you very much, Fidel. And thank you everyone who joined today. Thank you. Bye.